Good morning, my dear friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. I think this will be very convenient for the continuous study. My dear friends, in the last class we have discussed about the DMD and its incidents, pathology, etc. So today we shall continue with the genetic etiology and pathogenesis. You know that dystrophin protein, which is very responsible for maintaining the balance between the muscle fiber and as well as extra muscle environment. Okay, so that is get affected, and how it will going to lead to all these problems, that we will going to discuss today, so that the concept of muscular dystrophy will be very clear. Now DMD and BMD are basically, they pass through the X-linked recessive hereditary pattern. We know that about 30% of the case may be due to the new mutation. This is what we have discussed in the last day also. In 30% of the new mutation cases, mother is not the carrier. Yes, the female carrier state usually does not show any type of muscle weakness or other clinical symptoms. Suppose if carrier is there, she is carrier. Carrier means she will not show any symptoms. Okay, so a 427 kD cytoskeletal protein by name dystrophin, just a repetition, is encoded by the gene which is present in the 21st XP2 locus chromosome, in the X chromosome. In the P portion, again the locus is 2, second locus it is present. And actually this is a subsacrolaminal protein. What is this dystrophin? Dystrophin is basically a subsacrolaminal protein which is attached to the sacrolemma membrane and which forms the actin and myosin bands of the myofibrils. Okay, now let us see what is the importance. Now, attachment of this dystrophin with the muscle fibers occurs at 4 distinct areas or the domains. Now this dystrophin is attached to the muscle fiber, okay, protein, in a 4 distinct areas. Number 1 is, it is called as N terminus, okay, and then cysteine rich domain, then C terminus domain, like this. There are an N actin binding sites. So like this, 4 at the 4 areas. So from the extramuscular, that means what do you say, in the vascular department, from the vascular department it is attached to the muscles in these 4 areas, okay, like N terminus, cysteine rich domain, C terminus, etc. And this dystrophin is detected in the human fetus by 11 weeks. You can find this dystrophin protein in the human fetus after the 11 weeks. So dystrophin is a mRNA which is normally even detected in the cardiac muscles also, cardiac smooth muscles also, and more commonly it is seen in the skeletal and brain muscles also. So 3 important things are here. One, it is this dystrophin is a protein which is very important for the extra muscular compartment. That means from the vascular compartment and as well as to the muscle compartment. It is the connection between these 2 compartments. And how it is connected? It is attached in the 4 domains. Okay. Now this dystrophin is also found in the cardiac muscles. Dystrophin is also found in the skeletal muscles most commonly, and this dystrophin is also found in the brain muscles. So 3 things are in the brain. In the skeletal muscle it is present. Its absence in the skeletal muscle that usually leads to weakness, okay, loss of strength and weakness, whereas its absence in the brain muscles sometime. So even the brain muscles, brain related muscles are also undergo parenchyma, etc. They also undergo degeneration, and it may lead to intellectual problems, not always, may, intellectual problems and cardiac problems. Even it is found in the cardiac muscle. Therefore CPK-MB, okay, that is very specific investigation that we will see to see whether the dystrophin in the cardiac muscle is affected or not. Even the cardiac muscle is also affected or not. Find out that, we'll go for CPK-MB. M stands for myocardium. Now The molecular defect in the dystrophinopathies are of various types like intragenic deletions, point mutations of the nucleotide, etc. Now, what is the exact pathology? We shall see. Pathology occurs because of four reasons. Why this pathology start? Why the DMD start? Yes, dystrophin is absent, but why dystrophin is not functioning? But why the pathology of DMD starts? Muscular dystrophy starts. It is because of the four reasons. Number one, because of the abnormal functioning of the dystrophin, the connection between the vascular compartment and as well as the muscle compartment is get affected. Okay, and therefore there will be a defective blood supply to the muscles. Number one. Number two, there is defective nerve supply to the muscles. Okay. Number three, there is defects in the membrane of the muscle. There is a defect in the membrane because this dystrophin is also called as a muscle stabilizing protein. Okay, it stabilizes because it connects the muscle inside the muscle, internal aspect of the muscle to the external aspects of the nourishing environment. Okay, like vascular compartment, it connects. Okay, but when this connection is not possible, muscle is get deprived of the nutrition, muscle is get deprived of the blood circulation, and even this plays an important role in the nerve connection. Okay, so there is defect in the nerve supply to the muscles, defects in the membrane of the muscle, defects in the membrane like ultrastructure, increased level of the cholesterol in the membrane will be there. So membrane stabilities get affected. And lastly, because of the absence of this, the decreased activity of the sodium potassium ATPase pump. So these are the four reasons why the muscle is badly affected because of this absent or abnormal dystrophin protein. Because dystrophin protein connect between the internal environment of the muscle cell to the external environment in the vasculature, so that the nourishment will come, nerve supply will come, membrane will be get stabilized. Okay, sodium potassium pump will be normally working. Everything is depends upon this particular dystrophin protein. But when it is absent, the problem starts. So dystrophin is a rod-shaped cytoplasmic protein. It is a cytoplasmic protein. That means it connects the cytoplasm of the muscle with the external environment, nutritional environment. So this is a vital part of the protein. This is a very vital part of the protein complex that connects the cytoskeleton of the cell with the surrounding extracellular matrix, the nourishing area. What I am telling, extravascular. It's not exactly extravascular, extracellular. It connects the internal aspects of the internal environment of the muscle with the extracellular matrix. Okay, so therefore this whole thing is also called as dystrophin associated protein complex. Now, absence of the dystrophin failed to integrate the integral proteins and create gaps in the cell membrane. Now, because of absence of this protein, it fails to integrate the integral proteins, and as a result there is a creation of the gap in the cell membrane, and this leads to influx of the calcium ions. When the gap is there, through that gap the calcium ions start going inside the cell through the cell membrane. Hence there will be alteration in the signal pathways. This leads to alteration in the signal pathways and cause the water to enter inside the mitochondria. Okay, water inside the mitochondria. So the mitochondrial dysfunction leads to amplification of the stress-induced cytosolic calcium signals and stress-induced reactive oxygen species production. Now, in this cascade of changes, several pathways are involved and there will be increased oxidative stress within the cell and damages the sarcolemma. Sarcolemma is a binding where actin and myosins are packed inside. So eventually it leads to the death of the cell. Okay, because nutrition is not coming, nerve signal is not coming, and then more amount of water is coming, more amount of the calcium is coming. Okay, stress induced pathology is going on, oxidative changes are taking place. So actin and myosins are totally get suffered and sarcolemma is get damaged. Once sarcolemma is damaged, inside it actin and myosin is there, so they are get damaged, and all these areas get filled by some abnormal substance like fat substance, etc., which leads to pseudo hypertrophy, and eventually that particular muscle cell will go into die. So when this happens in almost all the muscles, most commonly it start with the proximal muscles. So muscle fibers undergo necrosis and replaced by the adipose tissue and other connective tissue, and which gives a bulky feeling to the which imparts the bulky feeling to the muscle, but actually there is no actin and myosin inside. So this is about the genetic etiology and pathologies. Now in the next class we shall see about clinical symptoms of DMD and BMD. Thank you very much.